Okay, let me just take the chair for a moment just to present him. Next talk will be given by Professor Mollenhauer. He's the director of the Nanomedicine Cancer Center of the University of Southern Denmark in Odense, and his talk is about meaning genome information for new starting points in personalized cancer medicine. Please, John, whatever you would like.
Yeah, thanks a lot. We switched to that schedule because we were already joking yesterday evening how I would introduce myself and have a dialogue with me. Yeah, I would like to introduce a little bit into links that we can draw between genomics disciplines and nanomedicine. And that corresponds very much to the approaches which we pursue. So just to get you briefly in touch with that issue, with nowadays technologies, we are in principle able to generate for each individual patient's tumor comprehensive molecular fingerprints that concerns genome-wide mutations in all 20,000 genes, which we can generate by next-generation sequencing. Still at considerable cost, but doable. And we can assess mRNA, microRNA expression patterns in the tumors. Taken together, this is already nowadays tried to be implemented in coming to a better prognosis and to a better prediction of therapy response and the optimal treatment. However, within these profiles, there's also much more information hidden, namely the information for novel causal cancer genes and mutations. So once you have been identifying them, you actually can go ahead and convert that into what we call synthetic lethal screens. That is where you scan for drugs that kill a cell that has a cancer-specific trait or mutation over a cell that lacks the trait. And normally this provides you with a very broad therapeutic window, so that would give rise to a preferential drug. Now, in our settings, we have one preferred drug format, that is nucleic acid-based nanodrugs. This is the simplest expression of such a nucleic acid-based nanodrug. It's simply a continuous nucleic acid molecule where the front part folds up in a three-dimensional fashion so that it can recognize a receptor on the surface of the cancer cells. So in a certain sense, it works like an antibody, but it's very much smaller than an antibody. And the hind part then would be the active drug component that exerts the synthetic lethal effect. In our case, this would be an siRNA. So we can stay within the nucleic acid world with this kind of design. But of course, there are alternatives to link this with liposomal technologies and other drug delivery devices. But I would like to concentrate rather on this first part here. So we have launched several projects, first instance to identify new causal cancer genes and mutations. This is one of them where we deal with a set of 140 genes that have been identified by next-generation sequencing as having potentially meaningful mutations in breast cancer. So this is a publication that is already quite some years ago. Nobody so far really cared of the gene set. In this project, it's actually larger than only these 140 genes because we generate or analyze the 140 normal gene variants versus 140 mutant gene variants, meaning one representative mutation as recovered by next-generation sequencing. So plus controls, this makes up a total set of roughly 300. So up to this time point, we have been analyzing a set of nearly 120 of these configurations. Yellow dots here are controls, blue dots are all the genes in question, red dots are positive control genes. So to this end, we came up with a set of 12 primary hits, some genes that promoted cancer growth, others that suppress cancer growth. And if I pick up these two examples here, you can see there's a good correlation of the effects that we see in vitro assays with the situation in the patients. So this growth promoter here, high expression levels indicated by the red curve are associated with a poor survival in breast cancer patients, while for this growth promoter, high expression levels are associated significantly with a good survival in breast cancer patients. I picked up two examples here. We have two genes, that is the data from the discovery screen, data from the validation screen, discovery, validation screen. You have always the wild type, the normal variant, indicated to the left. And then in this particular case, we analyze one mutated variant. You can clearly see this is a strong breast cancer cell killer, so to say. We know that it induces apoptosis. While if you use the mutant variant as identified by next generation sequencing, you have a loss of function. And that's also pretty good in accordance with the data we see in breast cancer patients. So high levels correlate with a good survival. So you can say that is for relatively sure a new tumor suppressor. Well, a very similar situation here for this candidate gene. The wild type variant suppresses growth. We have one mutant variant that actually has a loss of function again, that is deficient for growth suppression, and a second mutant variant that does not exert any effect, where the mutation doesn't exert any effect. This was also valuable information because this mutation has also been identified by next generation sequencing, but obviously has been a false positive. Also in this case, we have an association with a good survival prognosis in breast cancer patients, so this is also a tumor suppressor. Well, in another project, we dealt with a set of around about 100 genes and microRNAs, which we suspected to play a role in metastatic spread of breast cancer. Also in this case, we started with analyzing the effect of the genes on the breast cancer growth. Again, we have several candidates here which promote breast cancer growth, and some here which suppress breast cancer growth. But here in this case, we in parallel performed also a high throughput invasion screen, which is one of the steps in the metastatic cascade. From this screen, we identified two invasion promoters, one very strong, even better than our positive control, and three invasion suppressors. So I would like to pick up one particular example because that created a very interesting scenario also with regard to cancer stem cells. And this is this gene which we actually identified as a growth suppressor by our systematic assays and as an invasion suppressor, and where high levels are associated with a good survival prognosis in breast cancer patients. So from this you would say this is a pretty good candidate for a tumor suppressor. Now, of course, next we were interested in the mechanisms the gene would be involved in and the effects that it would elicit in cells at the molecular level. So we performed expression profiling on the cells expressing the gene. And to our big surprise, we could first see that it induces a hypoxia-like profile in the cells. So the top hits in the list when we analyzed for pathways were actually hypoxia-like pathways. That was surprising because hypoxia actually is very well known to induce breast cancer stem cells. Next, we found that it upregulates the stem cell factor, that's the transcription factor SOX2, which is highly expressed in cancer stem cells versus non-stem cells. That's shown here. Moreover, also SNAIL1 and SOX9 were found to be overexpressed in response to this gene. And these two genes have been shown upon concerted action of being able to convert mature differentiated luminal cells to stem cells directly. Next, we gained the information that if we look at the mammosphere forming efficiency of cells carrying this gene, and Simo already introduced you to that, that's a kind of substitute assay for scoring the number of breast cancer stem cells. What we found is that this is very efficient in increasing mammosphere forming capacity, as efficient as two very strong stem cell inducers, namely SNAIL1 and MYC. Moreover, when we looked at profiles, this is actually very similar to what Simo showed you before. This is a flow cytometric analysis, but we just put the data into a diagram according to percentage of the cells in the different fractions. Here we are using a different profile, that is the EPCAM CD49F profile, because that allows you to discern between different populations you also find in the normal mammary gland. So the first population here would be the mature differentiated memory cells, so to say. This would correspond to progenitor cells, so a little bit more primitive and at least bipotent. And this would represent the stem cell fraction. So what you can see, what is elicited by the effect of the gene is that the differentiated cells drastically decrease. We have a doubling of the progenitor cells and an increase of the stem cell fraction, at least according to this marker profile, from less than 1% to 30%. So this was very surprising because now we had a set of conflicting informations, a tumor suppressor at the one hand, but breast cancer stem cell inducer on the other hand. So we decided to let the in vivo experiment to make the decision for us. And in fact, when we implanted these cells into mice in an orthotopic implantation model and compared that to control cells just carrying a reporter, we found that tumorigenesis is significantly suppressed by this gene, and that can be monitored well by imaging, as shown here, and also by measuring the tumor volume. So, as it seems right by now, we have picked up a gene here that is a breast cancer stem cell inducer that functions as a tumor suppressor. So that is pretty much opposing mainstream. But I also told you that we wish to use such scenarios to convert it into differential screens. And we were in a lucky situation that within our center we could build up a large robotic high-throughput screening platform. In our settings, a differential screen would be 120,000 individual cell assays, which can be performed in around about 2 weeks, where most of the time is actually incubation time. The true capacity is even much larger if you go into smaller plate formats. So we decided, after we successfully set up screening already with adherent breast cancer cells, to dare the challenge to do screening directly in mammosphere cultures. Because as said, this is thought to reflect better the situation in the cancer stem cells, and mammosphere cultures are known to be enriched for cancer stem cells. So this is how it looks like. We use the negative control as RNA. You have spheres here all over, and if we use killing siRNA as a positive control, you can see that you actually don't have really spheres left. That's also reflected here, where we simply tested a set of negative controls, which all worked really fine, and 2 positive controls. We also have libraries available for microRNA inhibitors and microRNA mimics. To this end, we also set the screening up for microRNA inhibitors. So what you see here is are 4 different experimental conditions where we varied several parameters in cell seeding and medium composition, and 2 different microRNA inhibitor concentrations. But the main message is that we could robustly find a significant decrease of mammosphere forming activity with these 2 and with this third microRNA inhibitor here, regardless of which conditions we applied. So this looks very nicely robust to us. And we have just been starting with the first screen. So in summary, what I want to transmit in terms of a message is that we believe that genomics data sets can represent a really valuable resource to come to new starting points and new cancer genes. We have identified so far mostly tumor suppressors, which can be used or will be used by us to create appropriate scenarios for high-throughput screening, with the goal to identify siRNAs with synthetic lethality. MicroRNA inhibitor screening is working fine, and the pilot screen is on the road now, where we are pretty curious to see whether we can derive inhibitors that would have a cancer stem cell eliminating activity. But I also told you that we have 1 breast cancer stem cell inducing tumor suppressor. I don't want to be misunderstood. I do not think that this necessarily challenges the entire cancer stem cell concept, but I think it rather could mean that there is still another type of cancer stem cells existing which yet remains to be characterized. Well, I have to thank, of course, our grant givers. In first instance, the Lundbeck Foundation, which gives the major part of our financing to the center. The people involved in these studies, this is Helle Christiansen and Steffen Schmidt. They are involved in robotic screening. Ines Block and Caroline Müller are involved in the next generation sequencing gene set. And Angela Riedel and in part also Pernille, where do we have her? Here, have been involved in following up this one particular gene and in vivo studies. And you, of course, for your kind attention. Thank you.
Thank you very much. Certainly the idea of this tumor suppressor gene, which is inducing cancer stem cells, is really challenging. No? I mean, if we make the concepts very simple, then it's counterintuitive. No? So
yeah.
How can you induce cancer stem cells on the one hand and be a tumor suppressor gene? No? So how we explain this? I mean, that's the situation. We were discussing yesterday, and we didn't really reach any conclusion. but that's really challenging. So I was going to ask you one thing. Did you try to inhibit this tumor suppressor gene by siRNA in the mammosphere and see what happened?
Yeah, the issue with that is that it is expressed by very few, I think maybe only one of the cancer cell lines, breast cancer cell lines at all. So we have a little bit we are a little bit in trouble in finding a good model system for that. We believe that the reason is that it may require hypoxia in order to induce the gene. So we are working on that. At least in this one cell line, which has pretty low levels for the gene, we will give it a trial and try to knock it down.
We'll see what happens.
Yes.
Any further question, please?
Thanks for your talk. I've got two questions. The first one is modulation of the genes in cancer, and particularly the oncogenes, usually controlled by the microenvironmental factors. So to what extent these changes in microenvironment, let's say in a mouse, resemble those of the human? Maybe totally different genes are activated in a real cancer. And the second question is, coming back to the stem cell, very general question, where are they? How many are there? And how are we going to really hit them? Are we really moving too fast?
Well, that's a good set of questions. Maybe the last answer I can give you in some 30 years. But regarding the question of the cancer microenvironment in mice, it is acknowledged that it may be different, at least via using orthotopic mouse models. You have it in a more natural environment than using subcutaneous models. But there's also the possibility, I think, there are some models out where you can co-inject human fibroblasts, for example, to mimic the microenvironment. Cancer stem cells, how many are they, and can we eliminate them? I think you have to see that as a dynamic state. So it may differ from tumor to tumor, from very few percent to quite a lot, according to the present definitions by the markers. I think that's the main problem, that the markers that we have are useful in a way, but are not yet the end of the story. So, but they are in a kind of dynamic balance with the rest of the tumor cells. In the end, it will be the case, as we have already been discussing yesterday, that you probably have to hit both the normal cancer cells and the cancer stem cells in order to avoid that the former ones make a conversion to cancer stem cells back. But we will see what the future brings. It at least explains a number of issues like drug resistance, tumor recurrence, and from this point of view, it has already put forward cancer research quite a lot.
Okay, thank you. Let's move to the next speaker. Could you do the honors?